Greetings, and thank you for the opportunity to present at this conference. Joni and Melissa, it's really amazing what you're doing and heroic what you're doing for the women of Alaska, and a great honor to speak at this meeting. My talk today is on T-cell receptor gene therapy for cancer. It's my conflict of interest statement. Engineered T-cell therapy has had success with CAR T-cells in hematologic cancers, but progress in epithelial cancers has been more difficult and more research is needed in this area. And that's the main focus of my lab. In the short term, we are trying to investigate principles of cellular therapy in epithelial cancers using HPV-associated cancers as a disease model. In the longer term, we're trying to discover and develop cellular therapies for HPV-associated cancers and other malignancies, and we'll discuss that a bit today too. I want to start with some background and make sure that we are all clear on the difference between chimeric antigen receptors and T-cell receptors. These are the two main strategies for gene engineering T-cells to target tumors. So shown on the left here is a chimeric antigen receptor T-cell. And the chimeric antigen receptor engages its target through an antibody-like recognition domain, and the target is an antigen on the surface of the tumor cell. Unfortunately, only about 11% of protein coding genes are on the surface of the tumor cell. Most of the most attractive target antigens are actually located in the intracellular compartment, and this includes the E6 and E7 oncoproteins of HPV that we would be targeting in cervical cancer. To target those, we need a T-cell receptor, and a T-cell receptor targets a peptide bound to an HLA molecule, and the source of this peptide can be a protein anywhere in the cell. And this intracellular protein is processed into peptides by the proteasome, transported by TAP and loaded onto an HLA molecule. And that's what's presented on the surface of the tumor cell. And that's what can be targeted by a T-cell receptor. The rationale for studying HPV-associated cancers, particularly as a disease model for cell therapy for epithelial cancers, is based on three pillars. The first is the unmet clinical need. And of course, HPV-associated cancers, when metastatic, are essentially incurable. And this leaves us with a desperate need for better treatments. The E6 and E7 oncoproteins are very attractive therapeutic targets. They are constitutively expressed, they're immunologically foreign, they're derived from the virus, and they're not expressed by healthy tissues. And this is an almost unique constellation of characteristics and makes these really elite targets in epithelial cancers. And related to those very well-defined, constitutively expressed tumor-restricted antigens, there are unique research opportunities, again, just because these antigens are so well-defined. Our first efforts to try to target the HPV E6 and E7 oncoproteins with cellular therapy were with TIL therapy. And for this approach, from a patient with metastatic tumor, we operate and remove a tumor. We grow T cells from tumor fragments, generating multiple T cell cultures, and then expand these cultures and return them back to the patient with an intravenous infusion. With this approach, we found responses in 7 of 29 patients with a range of different types of HPV-associated cancers. Importantly, 2 of these patients had durable complete responses. That is, a single cell infusion caused complete regression of all of their cancer. And both these patients had cervical cancer, and then they're more than 8 years out without recurrence of their disease. And I'll show you more about that in a moment. This approach is being developed by Iovance Therapeutics with the drug name LN-145. We also did some interesting science around this. We were trying to target the HPV antigens with the TIL that we gave, but it turns out that in the successful treatments, there's targeting also of cancer germline antigens and mutated neoantigens. And I'll show you how we're also building on this finding of the targeting of other antigens as well. So this was the first patient to have a complete response to TIL therapy for an HPV-associated cancer, and really any epithelial cancer. This was patient 3, a 36-year-old woman with squamous cell carcinoma that was HPV-16 positive. She was treated with multiple lines of combination chemotherapy and chemoradiation in different settings. And she had the disease that's shown here by the yellow arrow. She received a single cell infusion with TIL, and all of her tumors went away. And the neat thing about this kind of treatment is that it's a one-time infusion. So in her case, where all of her cancer went away, she's been able to go about her life. And she's now more than 8 years out. This is a photo that she sent me a year or two after she was treated from the Kansas State Wrestling Championship, which her son won as a sophomore. And then she sent me follow-up photos when he won it as a junior and a senior. And then, of course, he went off to college. So it's been great that she's been able to go about her life and be a mother to her son after just a one-time treatment. This is patient 6, a 36-year-old woman who had a HPV-18 adenocarcinoma. This patient was treated with cisplatin and radiation. Her primary tumor was refractory, and she had pelvic and then extrapelvic progression, had complications from the cancer that delayed her treatment. And when we treated her, she had the tumor shown here with the yellow arrows. This little dot is a ureteral stent. We gave her a one-time TIL infusion, and all of her cancer went away. We were able to remove the ureteral stent. And she's more than 8 years out without cancer and doing great. This is a photo about a year or two after she was treated that her mother took. She didn't have any other family, so she got stuck in a photo with me. The TIL therapy has been informative and important. It's shown that cell therapy can mediate complete regression of cervical cancer, epithelial cancer for the first time. But it has some drawbacks. One is that it requires surgery. Another is that it takes some time to generate the cells for treatment. And another is that there's really no control over the cell product. Whatever grows out is what you have to give. So we've tried to overcome these limitations with next generation approaches. And here is the first gene engineering approach that we attempted. So from the patient with metastatic cancer, we obtained peripheral blood. The T cells from peripheral blood are gene engineered with a retrovirus encoding a T cell receptor that targets the E6 antigen. This is a T cell receptor that we discovered in the lab. We expand these cells and then we infuse them back to the patient. These cells target the E6 antigen by recognition of the E6-2938 peptide presented by an HLA-A2 molecule. With this approach, we saw responses in 2 of 12 patients, which is encouraging in that there were responses, but it's discouraging in that we were really hoping for a higher response rate. We also learned a lot about tumor intrinsic immune-related genetic defects, and I'll talk about that more with our more recent studies as well. However, when we started the E6 TCR clinical trial, we continued to look for T cell receptors in the lab and actually discovered a better T cell receptor that targets the E7 antigen. So what I'm going to show you next are our data from our E7 TCR cell therapy trial. And the E7 TCR treatment works like the E6. Again, it's a peripheral blood T cell collection, engineering with a retrovirus encoding, in this case, the E7 T cell receptor, expansion of the cells, administration of the cells. The E7 T cell receptor recognizes the E7 epitope presented by an HLA-A2 molecule. So the trial design was a first-in-human phase 1, 3+3 clinical trial. Treatment was with E7 TCR gene-engineered T cells. We call these E7 TCR T cells. Patients have to have a metastatic HPV-16 positive cancer, and they have to have the HLA-A2 allele. Any prior treatment was permitted, including checkpoint inhibitors, and 8 of 12 patients had a prior checkpoint inhibitor, 8 of 12 patients. The treatment was with E7 TCR T cells, and the dose escalation is shown here. starting with 1 billion, 10 billion, and 100 billion cells. We did not encounter any dose-limiting toxicity that prevented us from escalating, and we just stopped at 100 billion cells because that's about all that we can reasonably make. The treatment schema is shown here. The patients receive a conditioning regimen with cyclophosphamide or fludarabine. The cyclophosphamide dose is either 30 or 60 milligrams per kilogram. We've really moved now to exclusively using the 30 milligram per kilogram dose, which is much better tolerated and seems to be very effective. We give a single infusion of the engineered cells and then aldesleukin, which is dosed to tolerance. There are a lot of aldesleukin hatches here, but really we generally give the aldesleukin only for a day or two. And we've come to really stop the dosing when we see any concerning toxicity, and we're generally able to keep people out of the ICU or any serious adverse events related to the aldesleukin. So the patients treated on the phase 1 trial are summarized here. There were 12 patients. Sites of disease are the typical range of different HPV-associated cancers. The metastatic disease sites are generally extensive, multiple tumor sites and multiple tumors at each site. So these were very advanced cancers. And this is reflected too in the number of systemic agents the patients had received previously. And it is also reflected in the fact that 8 of the 12 had had prior PD-1 checkpoint inhibitors. Dose level is shown here and just reflects the dose escalation. The cells were generally very well engineered. You can see transduction efficiencies here, generally in the high 90s. The far right column shows responses. 6 of 12 patients had objective partial responses. The duration of responses is shown in parentheses, but really to appreciate the clinical activity, it's important to look at the specific patients. So this is the first patient we treated. She was a 49-year-old woman with vulvar cancer. She had previously received 7 agents. She had the disease shown here with these many lung nodules. This is all retroperitoneal, right pelvic and right thigh disease. She received a single infusion of cells, and many of these tumors completely went away. She ultimately ended up with a 8-month response. This is patient 5, a 59-year-old male with anal cancer who had had 3 prior systemic agents, and one of them was pembrolizumab, a PD-1 blocking drug. He had numerous tumors throughout his chest. These are all shown here with the yellow arrows, and some of this disease was quite bulky, like this disease here above his diaphragm on the right, and this large kidney tumor. All but 2 of these lung tumors completely went away. You can see the 2 small residual tumors here, and he ended up having a 9-month response. This kidney tumor shrank as well. And finally, this is patient 12, a 40-year-old woman with cervical cancer. She'd previously received 7 agents, again, including a checkpoint inhibitor, PD-L1 targeted drug. She had these 3 tumors. This one completely went away. This one completely went away. This one in the rectum shrank dramatically, and it eventually regrew a little bit. And I'll show you some of the science behind that in a minute. So we're trying to understand why some patients respond and some patients don't. And one thing that we've looked at is how well these cells persist in the patients after they're infused. With CAR T cells, it's often a very transient period of time when the engineered cells are present in the peripheral blood circulation, and then they become undetectable. But here it's different. We see high levels of the cells for a prolonged period after infusion. The color code here reflects the dose level. And at the highest dose level, the vast majority of all of the cells in the peripheral blood are actually gene engineered. And the same is mostly true for the intermediate dose. And there still can be easily detectable levels of these cells, even at the lowest dose. This is the frequency of cells in the peripheral blood that are gene engineered. And this is reflected too when you look at the concentration of the cells. So we've looked also to see if these cells are functional in the patients after they're administered, and this is an ELISpot assay performed on peripheral blood before and after infusion, testing for reactivity against the E7-1119 epitope. And especially at these higher doses, you can see that before treatment, the patients have very low or undetectable levels of E7 reactivity. And after treatment, they have very high, robust levels of T cell targeting of the E7 epitope. So we did a lot of research to try to understand response and resistance and looked at a lot of the characteristics of the T cells, and it was mostly unrewarding. What was rewarding, and I think where the key information comes, is from looking at the tumors. So this cartoon shows the findings in 3 of the 4 patients from whom we were able to obtain biopsies in the phase 1 trial. And 3 of the 4 patients had defects in the HLA complex that presents the target epitope. This is the E7-1119 epitope to the engineered T cells. So patient 5 was the patient with the extensive disease in the chest. He had a tumor in the chest wall that was biopsied before treatment that completely went away. He eventually had recurrence at a bony site, and the recurrent tumor showed this mutation in HLA-A2 that was not present in the tumor that completely went away that was biopsied before treatment. Patient 4 was a patient who did not respond to treatment, and a biopsy of that tumor revealed a premature stop codon in the alpha-1 domain of the HLA-A2 molecule. And patient 12 was the patient who had 2 out of 3 tumors completely go away, but the rectal tumor shrank and then regrew. The chest wall tumor that was biopsied before treatment did not show any defects in the beta-2 microglobulin molecule, but the rectal tumor, when it regrew, showed 2 important defects. One is loss of the start codon in 1 copy of beta-2 microglobulin, and the other copy of beta-2 microglobulin was completely absent due to a copy loss defect. So this is a terribly difficult problem that makes this tumor completely unrecognizable by any kind of T cell mediated attack. So I think we've gained a lot of insight into causes of response and resistance. And this slide kind of summarizes where we are. With the E7 TCR T cell therapy, dose was not limited by toxicity. Responses include complete regression of many tumors, bulky tumors, and PD-1, PD-L1 refractory tumors. There's high engraftment of functional engineered T cells. HLA and beta-2 microglobulin represent conspicuous biomarkers, and we're working on ways to try to identify problems with these molecules prospectively, which would certainly help with patient selection. We have a phase 2 trial that's ongoing at NIH, and we'll be starting this up at Rutgers Cancer Institute as well. And we're also looking to move this approach into an earlier disease setting, particularly with an induction therapy trial for local regionally advanced cancers, where this kind of approach may especially make sense because it's a one-time treatment that causes rapid tumor regression, and unlike other induction strategies, doesn't require cycles of chemotherapy that lead to increasing morbidity and complicate delivery of definitive therapy. I'm going to move on now to talk a bit about how we're trying to extend this engineered cell therapy approach to targeting other kinds of cancers through other types of antigens and lessons learned from targeting HPV-associated cancers with TIL. So this is patient 6, who we treated with TIL therapy, and she had a complete response. All of her tumors went away and they stayed away. We were trying to target the HPV antigens with her TIL, and when we analyzed the cell product that we gave her, it turns out that we were doing that. This gray wedge here shows you that 14% of the cells that we gave were a T cell clone targeting HPV-18E7. But interestingly, 67% of the cells that we gave in the top rank clone that we gave her in her TIL were all targeting another antigen, and it took some work to figure this out, but it's actually an antigen called KKLC1. This is a cancer germline antigen. So the T cell receptor that was mediating this reactivity is HLA-A1 restricted, and HLA-A1 is an allele that's present in about 25 to 30% of the population. This target antigen, KKLC1, is a cancer germline antigen, which means it's expressed in development, but silenced in mature cells other than germ cells. And it's reported by PCR to be expressed in a high frequency of gastric, triple-negative breast, and non-small cell lung cancers. And we found that it's also highly expressed in cervical cancers, like this patient. So we were able to capture the exact T cell receptor from this patient that was dominant in this response that appears to be curative. And we can then introduce the genes from that T cell receptor to anyone's T cells and use these to treat their cancers. So we tested this concept in an animal model, and that's what's shown here. This is an immunodeficient mouse model where the mice have either a 4156 cervical cancer cell line implanted on their abdomens. This is a cell line that we generated in the lab, or an A375 tumor line. This is a melanoma line that's commercially available. And when you treat them with engineered T cells targeting KKLC1, there's a dose-dependent tumor response. This is at the lowest, intermediate, and highest dose. And at the highest dose, all these mice appear to be cured. The same thing with this A375 cell line, except for that many of these tumors grew back eventually. And these cells are missing the KKLC1 antigen. We determined that in the laboratory. And when you look, the A375 cell line has heterogeneous expression of KKLC1, whereas the 4156 cell line has uniform expression. But based on these data and others, we're now moving this to a phase 1 clinical trial. So to summarize, the KKLC1 T cell receptor was the dominant T cell receptor in the TIL that cured a patient with cervical cancer. The KKLC1 TCR T cells mediate tumor regression in mice. And again, this supports testing this approach in humans. And a phase 1 clinical trial will be starting soon at the NIH, and shortly thereafter at Rutgers. We're getting close to opening that as well. So here is my final slide. I've just summarized what we've talked about today. HPV TIL have demonstrated safety and clinical activity in HPV-associated cancers. And this is important as proof of principle for cell therapy in an epithelial cancer with durable, complete responses seen for the first time. This is being developed now by Iovance Biotherapeutics, and hopefully it's an approach that will become widely available. The E7 TCR T cells, the gene-engineered cells, have demonstrated safety and clinical activity. These data are important because they provide proof of principle for engineered T cells in an epithelial cancer and, of course, in HPV-associated cancers. The phase 2 trial is ongoing, presently open at the NIH. We'll be opening that at Rutgers as well, and we're planning to do an induction therapy trial at Rutgers. The KKLC1 TCR, I think, represents a promising approach with the antigen, a promising target for a range of common cancers, including cervical, lung, gastric, and triple-negative breast. There are other efforts in the field that I think we should all continue to watch. These include approaches to target the MAGE-A4 antigen in a range of different types of tumors, and a recent report of TIL therapy in lung cancer, where TIL plus PD-1 was given after progression on PD-1 alone, and interesting results were observed. With that, I'll conclude, and thank you for your kind attention.
All right, thank you so much, Dr. Hinrichs. If you want to start your video. Oh, perfect. Okay, there he is. So as everybody gets up and going, I'm going to interject my usual question. What do you think about how this technology might change, or what sort of differences and responses you've seen with patients with prior PD-1 exposure? Because I really think, especially as it relates to cervical cancer, that we're going to see PD-1 usage make its way further and further up the lines of treatment. And I would guess that the majority of patients are going to be prior PD-1 exposed. And how do you think that that is going to impact this technology?
Well, that's a great question, and I think it's getting right at a really important issue. And the short answer is that consistent with other cancer therapies, treatments that rely on the same mechanism of action, and here it's a T-cell based mechanism of action, usually work worse after another agent that works through the same mechanism of action. And I think that is definitely going to be true here. And we're actually seeing it already in the TIL therapies for melanoma. There was a recent paper published that looked at TIL given at a single institution in different trials over a long period of time that spanned introduction of PD-1 based therapies and also targeted therapies for melanoma. And there is a definite drop off in response rates, and especially, unfortunately, in complete response rates as patients receive prior lines of therapy. Interestingly, not just PD-1, but other lines of therapy too. So I think that other immunotherapies probably have a negative impact on T-cell based, adoptive cell transfer immunotherapies, and that's starting to emerge.
And so as sort of a follow-up to that question, as this therapy makes its way to sort of the front line, the bench where it might be more readily available, do you think that there are certain patients that we should consider this before PD-1 or before, like, a patient comes with, say, a cervix cancer that's widely metastatic? Do you see a role for this to be sort of step one?
Yeah, great question. So right now all these treatments are experimental, of course. There's no FDA approval for the Iovance cervical TIL at this point. They have breakthrough therapy designation, but we haven't seen data from an industry-sponsored trial. And the clinical trials that I've done, are just that they're clinical trials. So none of this has made it to standard of care at this point. In terms of how I think these therapies should be developed, as they have to start in the last line just as a way to establish safety. And it's I think remarkable, especially now with all these patients receiving PD-1, as we just talked about, that we do see responses after PD-1 and fairly robust responses. And that's where you have to start. But I think what it's telling you is that you have to move them in terms of development to earlier lines for study. And so the next logical step would be move to first line metastatic, but it's really hard to conduct trials in that space because there's so many other competing drugs in that space, especially for someone like me who's trying to do this as an investigator at a single institution, very hard to get those patients. So the other place that we're looking to move this, or other way we're trying to get to an earlier line of therapy, is just with the small feasibility trial in the induction setting, where patients haven't received other therapies at all. And I think that'll show us a lot about what the true response rate is, and maybe point us towards more of a sense of true curative potential, if we can use it before the tumors are made really resistant. Does it make sense?
Absolutely.
Sure. Dr. Hinrichs, great talk. Thanks so much for all your information. And I just want to reemphasize, I think you are curing patients who are previously considered incurable, and that which is incredible and gives hope to all of us, really. And it's amazing being able to bring bench research to a place that is really clinically relevant. So again, thank you for all your work. I did want to step back a little bit for the benefit of our audience and put this in context. So a lot of my patients come to me and talk to me just about immunotherapy broadly. Can you kind of describe kind of the difference between what you're doing in terms of active immunotherapy versus passive immunotherapy. And also, now we're talking about cervical cancer therapeutic vaccines. And can you kind of put that all in context for us?
Well, that's a tough question. I'll do my best. So I think maybe we can look at 3 different types of treatment that are all immunotherapies that are probably what are on people's minds and what you kind of alluded to. So you can think about cancer vaccines, of course, checkpoint inhibitors, and maybe cellular therapies. And cellular therapies can be thought of in terms of both the TIL and the engineered cell therapies. And I talked about each of those last 2. So for the cancer vaccines, it's an interesting area of research, and it's something that's been going on for maybe 40 years or longer. It's been really tough to make cancer vaccines work in a way that is really convincing, where it's clear that there's a benefit to the patient, and especially what we've never really seen from cancer vaccines is you give them to a patient with a metastatic cancer and you reproducibly and reliably see robust tumor regression. So there's a lot of work still going on in that field, and we're learning more about target antigens, and there are different vaccine platforms coming along and combinations and different strategies to try to make that work. But right now we really haven't been able to get a good foothold with that approach. It's very attractive, by the way, because cancer vaccines in general are so low on toxicity, but they really have to shrink the tumors. The second modality is maybe the checkpoint inhibitors, which are really intended to take the brakes off of the immune response against the tumor. And they've shown some really amazing results, particularly in highly immunogenic tumors like melanoma. But still, the vast majority of people, even with melanoma, who have metastatic melanoma, get treated with a checkpoint inhibitor, go on to die of their disease. So I think it's important to have kind of a realistic, grounded view of what we've achieved as community trying to develop better therapies. Unfortunately, in cancers that are not melanoma, in terms of epithelial cancers, and particularly cervical cancers, it's considerably worse than melanoma. It's a relatively small minority who get a good response, and almost no one is cured. So they're great again in that the toxicity profile is pretty good, but the if you're a person who has a metastatic cervical cancer, you really don't have a lot of chance of being cured by that sort of an approach. The last and again, the way that it works is it relies on a natural immune response, an existing immune response in the body against the tumor, and just taking the brakes off of that response. The last one is the cell therapy approach, and that's what I discussed and what I'm focused on. And it has lots of disadvantages, including many that I mentioned. For example, for the TIL therapy requiring surgery, and these tend to be treatments that have to happen in the hospital and can have significant toxicities. But on the positive side, the TIL therapies, I showed you a couple of patients who have actually been cured by the approach. We think they're 8 years out from a single treatment with no cancer, so it sure seems like they are. The TIL, unfortunately, again, like the checkpoints, relies on the natural response of the body against the tumor. And we take those cells that are responding out of the body, grow them up into large numbers, and then give them back. And as I mentioned, sometimes it can work tremendously, but it fails more patients than it cures by a lot. So the final one, I think, the most promising path forward for really getting to a reproducible sort of and hopefully curative types of approaches is the gene engineered approach, where we again use the patient's own immune cells, but instead of relying on them to be naturally reactive against the cancer, which most of the time they're not, or at least not to the extent that we need them to be, we genetically engineer them to have reactivity. And it's not just random reactivity like what the next patient might have naturally, but it's the best reactivity we were ever able to find in years of looking for really good reactivity. When we find that reactivity, we capture the genes, and then we use that are mediating that reactivity, and then we use those genes for the T cell receptors mediating that reactivity to reproduce that reactivity in every single patient. And so I think that sort of an approach makes a lot of sense from that standpoint. And again, we're trying to move that up into earlier lines of therapy. Hopefully, we'll be able to cure more patients that way.
Thanks for that really extensive and answer. I think that helped put everything in perspective. So we got a question in the chat. How do you become part of a clinical trial? And as you alluded to, what you're offering is not yet prime time, right? So this is only in the auspices of a controlled clinical trial situation. So can you talk a little bit about what you're looking for in your patients, kind of what your inclusion criteria are?
So the active trials that we have going on right now are actually at NIH, which was my former institute. I've moved now to Rutgers Cancer Institute, and so I'm continuing to work with my colleagues at NIH, and they've kept these trials open, and we're doing translational research around them together, and we'll be opening them at Rutgers as well. The trial that's open now is a phase 2 trial for patients with metastatic HPV-associated cancers from any primary tumor site. So for gynecologic oncology, this would primarily be cervical, vulvar, and vaginal cancers. If you come across patients with anal cancer in your practice, also they would be eligible. It's any prior line of therapy is permitted. So like the trial I was showing in the phase 1, it's the same eligibility criteria. And the patients have to have the HLA-A201 allele, which is present in about 45% of people. It's more common in Native Americans, whites, and Hispanics, but we test everybody, and we've seen people from all races with the right allele. So that's the trial at NIH that, and I can provide contact information if anyone's interested in getting in contact with the investigators running that. The other trial that's opening at NIH, and hopefully the next week or two, is going to be the KKLC1 T cell receptor. And that will be for breast cancer, gastric cancer, lung cancer, and cervical cancer. And that requires the HLA-A1 allele, which is about 25% of patients. And that's a phase 1 trial, and patients have to have had 1 line of prior therapy. Is that helpful? And is that what we were asking?
Yeah, absolutely. I guess I also want to get a little bit to performance status and to toxicity.
Yeah, so, TCOG 0 or 1.
Yeah.
And yeah. And then, other things like, of course, they can't have, active infections, things that could make it dangerous for giving the conditioning regimen. The other thing that can be an issue is bleeding, because the conditioning regimen can transiently drop platelet counts.
You mean the cyclophosphamide and fludarabine?
Exactly. Yeah.
Okay.
Yeah. And then.
And then I guess I found it fascinating, because I didn't realize there's a lot of work being done on this, that your results really from peripheral blood were pretty close to from harvesting from tumor, given that you're able to do the engineering. And so do you think you'll be able to avoid tumor harvesting going forward?
Yeah. So we've stopped our TIL trials. So we're not doing that anymore. Everything's from peripheral blood now. Yeah. The one thing that people say a lot is they point to the 2 patients who we cured with the TIL, and then they say, well, you didn't cure anyone with the 12 patients we treated with the gene-engineered peripheral blood T cells. But those are really small numbers. It's very hard to make that sort of a comparison. And the other thing is that, the times have changed. And as Melissa, very astutely pointed out first thing, now most of the people are getting PD-1 treatment first. And that has to be hurting our chance of curing these people with other T cell based therapies. In fact, I think it's a little bit amazing that that they work at all, given that they're refractory to other T-cell based therapies at the time they get it.
All right, great. I've been, just a comment, a lot of the people that are watching this aren't used to the immuno jargon that we are. So let's just have to remember. So just for everyone. So I think one thing that I think some people don't understand, certainly I didn't understand until I got to know more about this topic, is, our immune system is supposed to be taking care of runaway cells, aka cancer, but of course we see cancer as the #1 killer of older adults. So, and a lot of that is that our regulatory cells get in the way. And I guess, can you talk a little bit about how important is then the induction treatment that you utilize? And I guess you're utilizing that no matter what, whether it's the TILs or the peripheral blood.
Yeah, well, that's a great sciencey question. That one's going to be hard to answer without jargon. So, the concept is that there are in tumors, in the body in general, a lot of inhibitory cells that are there to keep T cells that attack tissues from attacking normal tissues. And so there's a balance that's maintained by that, and that balance protects healthy tissues from immune cell attack, and it also protects cancers from immune cell attack. So one of the things we do to kind of disrupt that balance and try to get an effective cancer treatment when we use the cell therapies is give chemotherapy before we give the cells. And that chemotherapy is not aimed at the tumor, but instead at these inhibitory immune cells. And without getting into it too much, there are animal data showing that one of the mechanisms by which that conditioning regimen, that chemotherapy we give before the cells helps the treatment is by depleting these inhibitory cells. It works through a number of other mechanisms too. And it results in better proliferation, better activation, and better function of the anti-tumor T cells that we infuse. Studying that in the cell therapy setting in humans is really difficult. And the reason why the clinicians will understand, and that is that, you would want to look at the tumor not before you give the conditioning regimen, but after you give the conditioning regimen to see, what regulatory elements are present. And we just can't biopsy the tumor right after we give a conditioning regimen because it typically drops their platelets. And it's also at a point where they're soon also going to be at risk for immune infection. So from a safety standpoint, trying to biopsy tumors right after a conditioning regimen is something that I've at least so far just considered a non-starter because I just don't want to do harm to the patient. Right when starting the treatment, in particular. So I think that there's not as much known about that as we'd like to know. And then the other point I'll make, I'll try to keep my answer shorter, is that you'd like to biopsy these tumors too when they're regressing to look at in the microenvironment and see what's going on with regulatory cells and tumor infiltration and so forth. With the engineered T cells, we actually thought we were going to be able to do that in at least 2 patients where we got a biopsy of a tumor beforehand, and then they came back about 6 weeks later so we could get a post-treatment biopsy, but the tumor was gone. There was like nothing to biopsy. And so for 2 of the patients where we had like a perfect match, we weren't able to get the second biopsy again because the disease was gone, which was a difficult thing to explain to reviewers when we tried to publish the paper and kept getting criticized for not demonstrating what was going on in the tumor microenvironment after the treatment.
All right, well, I would like to thank Dr. Hinrichs for that incredible talk, and just use up one more minute of time just because the reason that I think that having people like you here is so important is because I have no idea about half of what you just said, just to let all of the patients know. You guys think that you're not sure what's being said. The G1 oncologist, really, the immunotherapy, the immune system in general is just, it's incredibly complex and takes people who spend their lives thinking about nothing but that to really help us navigate that. So I thank you for being that person. Can you quickly comment on what you think the role of this type of therapy, or just we'll call it engineered immune therapies, are for ovary cancer? There's a lot of patients on this particular conference that are thinking about ovarian cancer, and I know there have been some trials looking at, upfront vaccines, and I just wanted to end it with a quick, if you could just say what you think about that, that would be awesome.
Yeah, I think ovarian cancer is one where I feel like I should be able to figure out how to target it effectively, and I haven't been able to. And I feel like the field, and hopefully smarter people than I am, will be able to figure this out. Because it derives from a tissue where, in the situation that we're treating the cancer, we could actually kill the same cells from which the cancer derives, which is an advantage, because there should be an antigen or a protein that's specific both to ovaries and to ovarian cancer, and not to other tissues, that would make it a prime target, kind of like the CAR T cells for B cell malignancies. They have this marker CD19 on them. It's on normal B cells and it's on B cell cancers, and you can kill both and cure the patient. The patient can live. It just seems like there ought to be something like that for ovarian cancer. And so far, we haven't been able to clearly identify that. There are some targets that are sometimes expressed by ovarian cancers that are very specific to the ovarian cancer. They're thought to be cancer germline antigens, but I am a little frustrated by that. I'd like to figure it out, and I'm always kind of reading and looking for if someone's found a good target in ovarian cancer.
Well, thank you so much for that. I wish I could keep asking you questions, but unfortunately, I cannot. I want to thank Dr. Boyd for being such a great moderator and helping us navigate our way through all the questions, and Dr. Hinrichs and all of the other speakers for taking time out of their busy schedules to talk with us live up here in Alaska. Just can't begin to express our thanks.